Hello everyone. So today I'm going to explain you about the Pharmacognosy Unit 1 B.PHARM. Okay, as per the MSBT syllabus. So the topic one first is definition, present and scope of pharmacognosy. So let's start with the presentation. Right, I think you can see the first slide, that is I have written here the Pharmacognosy Unit 1, that is the definition, history and present status and scope of pharmacognosy. So today we are going to study the basic that is definition and scope of pharmacognosy. As you know, you are studying pharmacognosy, so it includes the plants and animal origin, marine source, all includes under the pharmacognosy. So we are going to study in this subject, that is the importance of the pharmacognosy. Okay, so now go to the next slide. So the flow of presentation for first topic is, first I'm going with the definition, then followed by that history, and then present status and scope of pharmacognosy. Same as it is, which is given in your syllabus. Okay, no changes are there. Now the definition, first we'll move to the definition. See definition. Pharmacognosy is the science of drugs of biological origin, that is plant, animal and mineral, like I said. Right? So pharmacognosy is the science of drugs of biological origin. Biological origin means it is mostly coming from the plants, or what we can say from the raw plants, raw material, animal sources, marine sources, like that. So biological origin. Okay. Then we will distribute the word pharmacognosy, like it is coming from the German scientist. Here you can see the word pharmacognosy was coined in 1815 by a German scientist, Sidler. Okay, Sidler. Remember the name Sidler, which has been derived from two Greek words. First is a pharmakon and second is a gignosko. Pharmakon means drug, gignosko means acquire the knowledge of. Okay, so this is the word which is divided into the two parts. Now we'll go to the next slide. We'll see that is very important definition, that is detailed definition of pharmacognosy. Okay, it can also be defined as the objective study of crude drugs of natural sources processed scientifically. Okay, so here now the most important definition I am going to highlighting here. So please remember, you have to write this definition in exam. Pharmacognosy is broadly defined as the scientific and systematic study of structural, physical, chemical, and sensory characters of crude drugs of vegetables, animals, and mineral origin, along with their history, method of cultivation, collection, and preparation for the market. Yes, so that is about the whole, we can say the whole combination of the pharmacognosy कि आपको structural study करना है, chemical constituent study करना है, then biological source study करना है, all those things combined, it is known as pharmacognosy. Okay, and please remember, you have to write the cultivation collection also, because that is also the important part, because you know we are studying the natural products or natural drugs which is originating from the crops and all. So cultivation collection is a very important step. Then next see, that is the crude drug. Crude drug is very important word, because mostly in cognosy we are going to study the crude drugs, all crude drugs. Crude drugs, their synonyms, their biological source, their chemical constituent, their uses, all those things. Okay, so simply generally if I will ask you what is crude drug, you can give me the answer. Crude drug means it is not processed, or it is not completely gone under the any what we can say procedure, that is crude drug. Exactly correct, not at all wrong. So see, it is a simple drug. Crude drugs are plant, animal, and their parts which after collection are subjected only to drying and making them into transverse or longitudinal pieces. Means when you will cut the plants, you will get the pieces, or you can cut into the longitudinal or transverse section. So that is exist in the natural form. It is known as crude drugs. Now the next, the whole sum of pharmacognosy is, it is a combination of botany, Like you have studied in 12th standard botany and zoology, two parts you have studied. So botany, ethnobotany. Ethnobotany, the meaning of this is study of traditional uses of plants for medicinal purpose. सो इथेनोबॉटनी का मीनिंग क्या होता है कि ऐसे प्लांट्स जिसका हम ट्रेडिशनल यूजेस लाइक मेडिसिनल पर्पज के लिए यूज कर सकते हैं, जैसे कि तुलसी हो गया, देर आर मेनी विनका हो गया, देर आर मेनी प्लांट्स लाइक डिफरेंट volatile oils, then glycosides. All those plants are having the what we can say, the medicinal purpose. Then marine biology, from marine biology also we will get many things like different essential oils, vitamin E and all those, right? Vitamin A from shark liver oil. So marine biology also the combination, that is also the part of pharmacognosy. Microbiology, herbal medicine, chemistry, pharmacology, pharmaceutics. That's it. This is the wholesome combo pack of pharmacognosy. Now, the next slide, we'll go for the history, okay, which is very important. So, let's come to the history. That is, those are the scientists you have heard about, the Shushruta Samhita, Hippocrates, Charak Samhita, right? So, those were the scientists who has discovered the plants and their origin and their medicinal purposes, we can say, right? So here I have put the images so that will fit in your mind. Yes, Charak has started in 300 BC. BC means before Christ. AD means Anno Domini. You can search the long form as well, but there is no need to write the year of those scientists. Just remember which are the scientists, कि कौन से scientists थे और उन्होंने क्या किया, क्या discovery किया या क्या found किया, right? Charak, Sushruta, Hippocrates, Aristotle, Dioscorides, and Galen. Galen is also known as father of pharmacy. Next, here is an image which I have directly like insert from the Google itself. You can see here that Egyptian Ibirus papers. They in that ancient times people were extracting the plants parts and they were making the kadhas and they were applying on the wounds and everything, right? So one image that will inspire you. Then this one also you can see in the father of like Hippocrates. So they were found the medicine for the like fungal infection in ear, and they were adding the kadhas and they were adding that what we can say solutions to cure the infection. So that is this is also one image for creating image in your head. Ki yes, like this they were treated at that time. Then next, Dioscorides. They have found many plants and they have found many uses of those plants. So it is also the most important, that is a materia medica. The materia medica, that is from the Dioscorides, which the scientists found 600 medicinal plants. So remember, see how the natural products are important for us. Then next, this is very important and very interesting slide. First, I'm going for the first scientist, that is, Hippocrates. So here you can see Hippocrates, the era was 62 to 370 BC, which deals with the anatomy and physiology, also known as father of medicine. Now, the next is, here you can see me also, but I am not the scientist. Next is, can anybody guess this? I think so, you can't guess this. So next is Aristotle. Aristotle, the scientist who has found the era 384 to 322, who wrote about the animal kingdom. Also Theophrastus in that time itself, 370 to 287 BC, wrote about the plant kingdom. So Aristotle and Theophrastus both combinedly find out two: animal kingdom as well as plant kingdom, and they have described the medicinal uses of plants. There are many medicinal uses of plants, right? Then third, everybody knows who is like, if you are watching from India or from any other country, Charak, right? Who has the most old, or what we can say most popular scientist at that time, Who has written Charak Samhita to get to know about this? Like Charak, you can see in third Charak in 3000 BC in Charak Samhita made 50 groups of 10 herbs to serve his physician need. Right, so he has found many groups of herbs which cure the different injuries. Then next we'll go for who is next. Any guesses? Yeah. Next is Sushrut Samhita. Charak after Sushrut. Sushrut also collected many herbs, that is 760 herbs in 7 sets based on common properties in Sushruta Samhita. Okay. So Sushrut scientists also, they have also discovered the many herbs which is very helpful for the medicinal purposes. After that next is the most favorite, that is Dioscorides, which they have written in Materia Medica, and they have also described the 600 different medicinal plants. If you want to see, you can search that directly, Materia Medica, the Materia Medica, you will get about information about that. And the last one is Galen, which is the first pharmacist you have studied in jurisprudence subject or in many other subject, that is the Galen is first pharmacist who all remaining scientists has discovered the medicine, but Galen, who described the methods of preparing drugs of vegetable and animal origin and laid down many formulas, which is helpful for the thesis. Okay. So now the next present status and scope of pharmacognosy, we'll go for it. See here. Just give me a 1 second break, 1 minute. Yes, so sorry for disturbance, like 1 minute. So see, now the next is very important, that is a scope of pharmacognosy, right? So here you can see, first we'll go for the isolation of phytochemicals, that is glycosides from digitalis leaves. So there are many categories, student. Okay, so when you will study the pharmacognosy, at that time you will come to know what are the like categories: glycosides, alkaloids, there are anesthetics, there are many categories: volatile oils, resins, tannins, and every category I'm going to discuss with you in future videos. So please keep watching my videos and give this thumbs up and subscribe to my channel, right? So I definitely know you are going to do that. So I will continue with my next part, that is, see, isolation of phytochemicals. So in simple language, I will explain you this. That is, wait a minute, I will, yeah. So see, isolation of phytochemicals, the meaning of this word is, when you will extract the plant parts, and when you will, like what we can say, take the raw materials, you have to take those raw materials, and those raw materials you have to do the extraction of all those plants. So you will get chemicals. You can isolate chemical constituent from those plants, which will give you the activity. सो कैसे रहता है प्रोसेस, जैसे कि अभी अब डी फार्म हो गया, डी फार्म होने के बाद आप बी फार्म करोगे, बी फार्म होने के बाद आप एम फार्म करोगे, राइट? सो एम फार्म में आपको प्रोजेक्ट करना पड़ेगा. तो प्रोजेक्ट में कैसे रहता है कि यू हैव टू चूज एनी प्लांट्स, like if you are going for the pharmacognosy subject, then you have to choose any plant and you have to do the isolation of chemicals. So how you are going to do that? There are many procedures, starting from the distillation, then isolation, and then many things are there. Okay, so make sure that you are going through the extraction procedure and isolating all the phytochemicals properly, right? So this is very important process of the isolation of phytochemicals, which is the most important scope of pharmacognosy nowadays. People are finding out many plants, like, see, we have seen the history, Galen, Charak, they have also find many medicinal plants. But nowadays, modern research, you need, because every drug has many side effects, many adverse effects. So you need to make sure that that plant is having or the chemical which you are isolating from the plant, having the less side effect, less adverse effect, and which will be helpful for cure the patients, right? So that is the modern scope of pharmacognosy. So let's share my PPT again. Here you can see the next is like I said, right, like I said, natural product as a model for synthesis of new drug. Okay, so here you can't see me, I guess, right? Why? So natural product as a model for synthesis of new drug. Exactly. So nowadays people are using natural product because allopathic drugs are having a lots of side effect and adverse effect on human body. So the natural products are important for that. Then next is biosynthetic pathways investigation. There are many biosynthetic pathways investigation which will help for the preparation of the metabolites. Now, the next scope of pharmacognosy is drugs of direct therapeutic use. There are many drugs. Sorry, which you can use directly for the many uses. Like there are many steroidal drugs which is originated from the plants. Then vincristine, vinblastin used as an anti-cancer drug. Then there are antibiotics which you can use, those are also very much helpful and giving you the therapeutic uses, right? Then cultivation and collection of medicinal plant. Then the scope of pharmacognosy, you are doing the cultivation collection properly, like highly narcotic drugs, opium, cinchona, senna, clove. So opium is narcotic drug, cinchona, senna, clove, or other category drug. Okay, that also you are doing the proper cultivation collection of those drugs to bring it into the market, right? Then preparation of herbal formulation. This is the most like a common and most important part, because herbal formulation which Ayurveda, which cure the cause of disease. Nowadays people are moving towards the Ayurveda. Why? So you give me answer in the comment box. What is the importance of Ayurveda? Write in the comment box and let me know what are your views. I am going to say that Ayurveda is future of our generation, like a future of generation, because allopathic drugs are creating many adverse effects in our health. We have to take care of our body, right? So, and Ayurveda, the main use of Ayurveda, which will cure the cause of disease. So if you will cure the cause of disease, then automatically disease will go away. Then we will see what are the categories comes under the herbal formulation, that is Bhasma known as oxides of metals, then quad, that is extracts, then gutica, that is pills, lab, that is ointment, asava, arista, alcohol containing liquids, powders, medicated oils. So all those are the part of the herbal formulations. And one more thing here I want to say, that definitely you are going to study these things in detail also in separate unit. Okay, now the most awaited slide, that is the last slide of this presentation, structural activity relationship. This is very important. So see, I have put here two drugs, tubocurarine and toxyferine, which is isolated from the curare plant. So which is isolated from the curare plant, and this tubocurarine and toxyferine, both drugs are used for the muscle relaxant properties. Means if you are suffering from any muscle like in muscle contraction or something like that happening to you, then it is used as a muscle relaxant properties. And why it is used as a muscle relaxant properties? Because after isolation, like I said कि आपको कैसे करना पड़ता है, plant लेना है, extraction करना है, chemical separate करने हैं, and then you can use for the like a therapeutic purpose. Here, when from curare plant they have separated tubocurarine and toxyferine, so that time they have found out these both drugs contain the ammonium groups, and which is very helpful for the muscle relaxing property. So that's why SAR, that is structural activity relationship, is also the important factor of pharmacognosy. The hypotensive and tranquilizing action of reserpine are due to the trimethoxybenzoic acid. And then one more drug is there, that is reserpine. Reserpine you will get from the Rolfia, and this is also the important drug of the different category, like alkaloid category, and which give you the hypotensive and tranquilizing action. Means it will give you the calm and relaxed. Okay, so yeah, that's about it. And the question bank here, they can ask you the question bank like a defined pharmacognosy. From this unit, they will ask you the 3 type of question in exam, in MSBT paper. Define pharmacognosy, explain the history of pharmacognosy, and detail about the scope and present status of pharmacognosy. Okay, so prepare these questions well. And if you have any doubt, please put in comment box. I will definitely get back to you. Okay. Yeah, so that is about the question bank. 3 questions, 5 marks, and 1 question is for 2 marks. So today's course, that is the way to get started is to quit talking and keep doing. So always, you know, always quit talking कि मैं ये करूँगा, मैं वो करूँगी, मैं ये करना चाहता हूँ, मैं वो करना चाहता हूँ. So just leave all that and start doing whatever you want to do in your life, right? Thank you very much for patient listening. If you have watched the complete video, I hope you have watched the complete video. And please subscribe to my channel, that is Running Pharma by Priya. I have just started, so I need your support. Subscribe my channel, share with your friends, and give big thumbs up to my channel, right? And if you have any query, right? So if you have, sorry. If you have any query, any doubt, feel free to reach to me. I have put my email ID there. You can go in my bio and check their email ID is there. You can message me on email ID. I will get back to you. And please subscribe to my channel because I am preparing the presentations on many things. Like I am going to prepare the presentations on the job profile as well. What to do after D.PHARM C, what to do after B.PHARM C, and all those things. So thank you very much once again. See you.